So what I wanted to do today is talk a bit about evidence-based approaches to environmental chemical assessment. So translation, evidence-based approaches is systematic review. So you've heard a sort of a preview of that. So I work within the integrated risk information system within EPA, and we do chemical assessments. Our assessments, the result of them are toxicity values. They would focus on cancer and non-cancer. We're in the Office of Research and Development, so ours are technical assessments. Our assessments don't actually have an impact until they're used by program offices and thought about in the context of other information, exposure, etc. So these are just some of the laws and regulatory decision making. So this would be sort of how our assessments would get used in these kind of contexts. So we do our assessments using systematic review approaches. So a systematic review is essentially a study of studies. So when you think about an experiment and you think about an experiment has a methods section, that's kind of what systematic review does for literature analysis. It brings that level of transparency to analysis. And so they've really taken off in the field because it puts a lot of premium on being sort of clear about the methods. How did you identify studies? How did you evaluate individual studies? How did you look across studies to reach your conclusions? That's part of sort of standard features of a systematic review. You know, as Katja mentioned, they really had its origins in clinical medicine, but I think has really infiltrated literature analysis, period, across many different disciplines. And you know, now even in the journal publication world, it's probably harder to have a review published if it's not speaking to some aspect of systematic review. So here's just a schematic. So in the context, and I think this might use different phrases than what Katja used, but I think very conceptually, sort of a different flow of information. I'm just focusing on the top panel here. When you start a systematic review, you think about it, what is the question? What is the decision-making context? That would be scoping. You also do problem formulation. Has somebody else already done the assessment? Can I build off of somebody? Do I have to start from scratch? So a lot of these kind of decisions. And then when you think about the decision-making context, then you really start to think about what kind of evidence do you need to bring to bear to address the question. So you have this whole process of really trying to focus your question. And through that process, what you do is it starts to set up how are you going to find studies that address that question. And so then there's literature searching to find the evidence, and then there's coming up with screening guidance, criteria to sort of indicate when is a study relevant or not. And the idea in systematic review is that your criteria for study eligibility are transparent and clear enough that somebody else can understand them. Somebody else could screen studies and understand why it was included or not. So again, sort of this focus on transparency. In the IRIS context, our assessments tend to be, very broad. So it might be, what is the effect of chemical X on health outcomes? And those questions can be they can lead to very, very large evidence bases to evaluate. So we typically go through an approach where we might inventory what's out there and then make some decisions about where do we prioritize, especially in the case where there's a lot of evidence. So that's what's referred to as literature inventory and refining the analysis plan. And then you would go through a process of study evaluation. And again, this is really focused at the individual study level. There are a variety of tools depending on whether you're talking about RCTs, whether you're talking about human observational studies, in vitro studies, animal bioassays. Once you get kind of a better sense now, you understand sort of the extent of the evidence, a bit about the quality. Then in the IRIS process, we start to have more specific decisions about how we're going to organize our review. And then what I really wanted to focus on today, I think one of the challenges is sort of this process of a data extraction. I'm going to sort of spend most of my time in the following slides walking you through that, but it's one of the most expensive, most tedious aspects of doing a systematic review. And what I'm talking about there is I'm talking about now that you've got your studies, you need to summarize the design, the methods, and the results from those individual studies. And it takes high quality data extraction, probably takes 1 to 3 hours per study. And it's typically done by people who are making maybe $75, $100 an hour. So it's incredibly incredibly expensive. And so I think within the field, we're always looking for opportunities about how do we make this more efficient. And there's two flavors. One is, can we take advantage of data extraction that's been done by somebody else? And then that trips into conversations about database interoperability, trying to encourage the field to actually use a structured way for extraction. Not too long ago, the standard was, I just write it up in a paragraph or a word table, which is really hard to reuse. Okay, so one flavor for extraction in terms of efficiency is, how do I better take advantage of what's out there in the field, including if Katja does extraction through her process, and I have a question that needs those studies, how could I take advantage? The other flavor is thinking about, how can I electronically capture information from studies? Does this all have to be manual? So those are the sort of the two prongs to the challenges that are being explored. And then once you have your information extracted, then evidence synthesis and integration, like Katja, I use an approach I'm not going to get into now for time, that's sort of very much modeled on a GRADE approach, which is essentially thinking about many of the same considerations that you always think about, consistency, the quality of the studies, evidence of dose response, and trying to characterize those factors into a conclusion that describes your confidence in your findings. So that in the context of systematic review, that's referred to as a structured framework. And again, there are many flavors of it, and there's nothing really new. Many of them are sort of kind of based on Hill considerations, but I think what's happened is there's more attention to how do you operationalize that. So consistency, okay? So when do you describe in a transparent way what pattern of finding you think are consistent, not consistent, something in between? Okay, so I mentioned a bit again that I want to focus on data extraction. See if I think I've covered all that. Okay. So I'm going to talk a little bit about software application that we use within the IRIS program, others use, that helps facilitate this very painful process of data extraction. I'm going to focus on it because I do think it is probably the best thing out there for this application. It's web-based, it's open source, free, and has sort of structured interfaces to collect information on epidemiology studies, everything from controlled trials to case controls, animal bioassays, in vitro studies. Doesn't do high throughput or omic kind of stuff. It was developed by Andy Shapiro when he was at UNC working with Yvonne Rusin. I guess some of the other reasons why I want to, and again, I don't want to do a sales pitch on this application. It's just very convenient, I think, to sort of showcase some of the current workflows and maybe the opportunities to have breakthroughs on this painful process of extraction. One of the other features, and I'll show this in some of my follow-up slides, is that once you've extracted the information, then what Hawk does is it has nice ways to help you visualize patterns of findings across studies in a way that lets you sort of see information, but also use sort of through the web-based format, click to see more, so you can drill down and see more of the methodological details. So here's an example. This is sort of a data extraction field. This is what the extractor would see. This would be animal bioassays, and I don't know if you can read the content, but you're looking at things like the chemical name, the purity, the vehicle, the diet, if there was guideline compliance. We also have similar modules for collecting information about the animal model, species, strain, source. So I want to showcase this because these discrete pieces of information, in terms of the two-pronged attack, these are probably the parts that seem like they might be most amenable to electronic extraction. Electronic data extraction is incredibly hard, but the more discrete the information is, at least it's a little bit easier to work with. We also have an endpoint. So now we're getting sort of maybe into the second one. So, within Hawk, we have maybe 5 ways to sort of categorize an endpoint: the actual name, the system, the organ, the effect subtype. And as an extractor, what you find is sometimes you need all of these fields to describe an endpoint, and sometimes you don't. This is the part where, it's free text, in part this idea of trying to have a controlled vocabulary drop-down option maybe can work for system and organ. It really, really falls apart when you're talking about the endpoint level. I mean, just trying to think about how would you do that when you're trying to characterize endpoints in the published literature. Very, 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 varied space there. So this would be the part when, in terms of trying to tee up some of the challenges, is probably one of the harder parts of thinking about database interoperability, is how can we try to get the field to try to be a little bit more similar in how we summarize the endpoints. If we can start there, then we can have traction on the database interoperability. So here's an example of some, this might be, for example, a figure that you would see in the assessment. In this case, you're looking at, I don't know what you're looking at, an epidemiology study, chloroform birth defects. So this might be the visual that I mentioned. You can sort of look at patterns across findings, but then you can click on something and click to see more. So again, this is up to you how much detail you want to put in there, but this is sort of one of the carrots of using this kind of approach, is that you have it there and your reports get more concise, but you don't lose the detail. Here's animal evidence, the same kind of thing. I can sort of drill down a little bit here, and I can sort of see more about details of the results, the methods. I can see more the specifics of the number of animals in a group, the effect level. I can see information about, how the study was evaluated during study quality, including not just the rating, but the rationale. You can also, once you have it in this format, you can also display it in different ways, effect size format. So you can move away from just the no AL, low AL format. Here's another example. Again, this is sort of from the same underlying data extraction content. In this case, I think it's a nitrophen example in Hawk, but what you have is a collection of dose response across all kind of outcomes, everything in here. And the idea is that you might click on one that you find intriguing, and then you get the point. You can sort of see all that underlying data. So this is sort of what happens when you begin to encourage the field to collect information in this way. It's more accessible. It's easier to sort of look at patterns, and it's shareable. So in our process, when we finalize an assessment, then we would make available all the extraction in a way that you could, if you wanted to do quantitative analysis on it, then it's set up in that way. So I think in terms of the data extraction needs, I think I've mentioned this. So the two prongs are to think about opportunities for interoperability with other databases. There are some out there. They might not be developed or used in the systematic review context, but for example, ToxRef database, that's one that has animal bioassay information. It's conceivable that we might do an assessment and want to take information from that database and bring it into ours. So that's one example. I think some of the barriers is that some of the databases out there don't collect information in the granular way that I showed you. And so it makes it a little hard to crosswalk them. There's no consensus on the terminology, and this is a space where people can talk about trying to develop ontologies, but at the same time you have here and now assessment. So it's not like the field is just going to go on pause until we develop an ontology, right? So we need to sort of think about how do we keep going, but look for opportunities to at least match up the terminologies. Electronic data capture for full text, it's really, 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 really hard, even for the simple content, and especially when you're thinking about trying to get content that might help you understand study quality. Now you have to sort of interpret sentences that might not even be in the same paragraph. And also one of the barriers is that I think there's actually a very keen audience of modelers and programmers. What they need are training and test sets. And so, but it's not as simple as saying, here's a PDF, here's the information I extracted, have at it. What we found is that they actually need the full text annotated in a specific way that they can build a model around. And that annotation is not as simple as a comment box in an Adobe PDF file. So there would have to be a front-end investment, painful, to develop these training and test set that would kind of cover the space of what's happening. NTP has actually done some of it. I think they have like 100 studies now where they've used sort of a specialized software application to annotate them in the correct format, and they're focusing on the animal bioassay, more of this discrete content, strain, species, chemical. I think that's all I have. Okay.